Hello, everyone, and welcome to the Surveillance Advances Series' 2024 to 2025 season. This seminar series is co-hosted by the Public Health Agency of Canada and the National Collaborating Centre for Infectious Diseases, or NCCID. The title for today's seminar is Novel Partnerships and Approaches for Mosquito Surveillance. My name is Robert Sager, and I am a research staff at NCCID, and I will be your moderator today. The NCCID is funded by the Public Health Agency of Canada to provide knowledge and evidence for use in public health planning, programs, and policies related to infectious diseases. We are very pleased to be co-hosting this series with the agency. A quick disclaimer, the views expressed today do not necessarily reflect the views of the agency. I am joining you today from the city of Hamilton, situated upon the traditional territories of the Erie, Neutral, Huron-Wendat, Haudenosaunee, and Mississauga peoples. This land is covered by the Dish with One Spoon Wampum Belt Covenant, an agreement between the Haudenosaunee and the Anishinaabe to share and care for the resources around the Great Lakes. Today, Hamilton is home to many Indigenous peoples from across the Turtle Island. We honor the Indigenous peoples who have lived on and cared for these lands for generations. We are grateful for the opportunity to share and call this place home. The Surveillance Advances seminar series has welcomed participants and presenters from coast to coast to coast since our inception in September 2023. While surveillance is the most common term used in public health to describe the process of tracking health events and determinants through the collection, analysis, and reporting of data, due to concerns over its connotations, some have suggested the use of other terms, such as public health assessment. We acknowledge that there is still an ongoing dialogue about the use of the term surveillance in public health. For the time being, without a widely accepted term in the literature, we would like to note that we use the term surveillance in the series title to refer to the assessment of and response to public health threats as a core function of the public health systems. Now, before we dive into today's seminar, there are just a few housekeeping items I would like to mention. First, a recording of the seminar and presentation slides will be available shortly after the seminar on the NCCID website. That's nccid.ca under webcasts. Second, the chat box for participants has been disabled for this session. The hosts will use the chat box to communicate additional information with you. If you have any technical problems with Zoom during the seminar, please email us at nccid@umanitoba.ca. And finally, please send in your topic-related questions through the Q&A tab, and we will try to address as many as possible during the discussion period. You can like any of the questions to push them up in priority. We would also like to note that Surveillance Advances is a Section 1 self-approved group learning activity as defined by the Maintenance of Certification Program of the Royal College of Physicians and Surgeons of Canada. The seminar series is also approved by the Council of Professional Experience for Professional Development Hours for members of the Canadian Institute of Public Health Inspectors. If you require a letter for your participation today, please look out for the survey that will be shared after the seminar. And now I would like to introduce and welcome our speakers. Dr. Victoria Ng is an infectious disease epidemiologist and a senior scientist at the Public Health Agency of Canada. She completed her PhD in epidemiology at the Australian National University in 2010 and a postdoctoral fellowship at the University of Guelph from 2010 to 2012. Prior to joining the Public Health Agency of Canada in 2012, Dr. Ng worked for the Hospital for Sick Children and Public Health Ontario. Her expertise in quantitative risk assessment, disease prioritization, and statistical mathematical modeling of infectious diseases, particularly the transmission and expansion of exotic mosquito-borne diseases and exotic mosquito species in Canada under current and projected climate change. Dr. Ng is passionate about using alternative surveillance approaches to complement traditional surveillance and the use of citizen science to engage communities and leverage resources to keep Canadians healthy. Also joining us is Dr. John Soghigian, whose research focuses on the causes and consequences of parasite and vector diversity. He uses a range of genomic tools and evolutionary methods to understand why some vector arthropods are so problematic, while others, often their near relatives, are benign. John completed his PhD at Clark University in 2016, where he focused on coevolution and host parasite associations in mosquitoes. He then completed postdoctoral training first at the Connecticut Agriculture Experiment Station and then at North Carolina State University. where his research shifted towards using genomics to understand the evolutionary origins of species and phenotypes in arthropods of veterinary and medical importance. He joined the Faculty of Veterinary Medicine at the University of Calgary in June 2021. So thank you so much for joining us today, Victoria and John. Victoria, the floor is yours.
Great. Thank you, Robert. Thank you for that introduction. And I'm about to share my presentation. And I will have it in presenter mode. Okay, does that look good for you? Okay.
Yeah.
Perfect. I'm trying to, do you see the box with my face in it? I'm trying to move that away, but if no one else can see it, that's not a problem. Okay, I'm going to try to minimize it. Okay. Okay, well, thank you so much for allowing me to have the forum today to present. I've been given about 20 minutes with some time for Q&A, so I may breeze through these slides really quickly, but please feel free to ask questions at the end. And of course, I'll make myself available as well offline if there are additional questions that can't be answered. So my talk today is titled Scientists and Students Unite, a Citizen Science Approach to Mosquito Surveillance. And before I begin, I do want to acknowledge where I'm situated today. So I'm presenting to you today from my home in Oakville, Ontario, and I would like to acknowledge that I am on the traditional, ancestral and unceded territories of the Anishinaabe, Atwandarak and Haudenosaunee people. The town of Oakville is located on the treaty lands and the territory of the Mississaugas of the Credit. I have no conflict of interest to declare for today's presentation. And these are my 3 learning objectives. So the first one is I'm hopefully going to try to translate to you the importance of exotic mosquito-borne diseases or exotic mosquitoes to public health in Canada. I'll talk a bit about the limitations of traditional mosquito surveillance and then some alternative surveillance approaches with benefits beyond data collection. So before I begin, I wanted to do some icebreaker questions and I wanted to poll the audience to see how much you know about mosquitoes. So just 3 quick poll questions. The first one is, how many known mosquito species are there globally? So I think all of you should see on your screen that there's a poll question titled Poll Question 1. And I invite you to click on one of the answers. So it could be less than 10, 10 to 500, more than 500 to 2,000 or over 2,000. And I'll give you about 20 seconds to respond. And don't worry, no one's linked to their responses. We won't know if you answered correctly or incorrectly. I'm not sure if the moderator can put, is there a countdown to when the poll ends?
No, we could just close it when you're ready.
Okay, perfect. Okay, so we have the majority of people, 38% of you responded to more than 500 to 2,000, which is really close, but it's actually over 2,000. So in fact, there's approximately 3,500 known mosquito species. I emphasize known because we obviously don't know that we know all the mosquito species that are out there, but there's over 3,500 mosquito species globally. Okay, and then I'm going to advance my slide to the next question, and I think that will also coordinate a poll question to you. Okay, so what proportion of known mosquito species from feed on humans? Is it A, less than 35, so 1%, less than 1%? Is it 6%, about 200? Is it 10%, about 350? Or is it 50%, about 1,750? Oh, I think we might have poll question 3 on the screen. Oh, okay. So I see poll question number 2 on the screen. If you wanted to sort of check off a box in 20 seconds or so. Okay, I feel like the time's going to come up soon. Okay, so most of you thought 6%. You're spot on, actually. It is about 6% or about 200 species that are known to feed on us. So really it's very small compared to the number of known mosquito species there are. And in fact, most of them prefer to feed on plants or animals, which is good for us. Now I'm going to move on to the next slide. Third question and last one, of the small number of mosquitoes that feed on humans, so 200 or so, how many can pass on pathogens that can cause diseases in humans? Is it about 10%? Is it 30%, 50%, or 100%? Okay, there's the poll question. So 10%, 30%, 50%, or 100%. Okay, so many of you think that it's either 10% or 30%. It is actually 50%. So approximately 50% or about 90 species out of 3,500 species. So it's not that much. So I'll close the poll and I hope you had fun playing that game. Let's advance the slide. So you would think, based on the questions and the polls, odds are for us, right? There's lots of mosquitoes, but most of them don't feed on us, and only half who do feed on us can make us sick. Unfortunately, we still get about thousands of people in Canada that get sick every year from mosquito-borne diseases. And the reason is it's really, while the odds are for us, there's just so many mosquitoes out there. I think I read somewhere for every person on the planet, there's about 16,000 mosquitoes. And so, you know, when you look at the odds the odds are probably in the favor of the mosquitoes. And so thousands of people do get sick in Canada from mosquito-borne diseases, and they can get sick either from diseases that are circulating inside of Canada. So you've probably heard of West Nile virus. Less common, you've got California serogroup viruses or Eastern equine encephalitis. And then you've got the exotic pathogens, so those that don't circulate in Canada, like dengue and chikungunya and Zika that are occurring in other countries outside of Canada. But a lot of Canadians actually travel, and when they do come back, they present with the diseases that they've acquired overseas, and that is part of our healthcare system when they come back to Canada. And so I wanted to focus next on my presentation on Aedes albopictus. It's one type of mosquito species out of the many that can cause illness in humans, and they're responsible for transmitting exotic pathogens in humans for Canadians. So I call them exotic because these are diseases that are not currently circulating in Canada. So for example, dengue, chikungunya, and Zika virus. So Aedes albopictus is a mosquito species that has a very distinct feature. So you can see on the legs and the body, there's a black and white stripe mark, and that's a distinguishing feature of Aedes albopictus. And this particular mosquito species wasn't present in Canada prior to 2016, but it was first detected in 2016 in a very small part of Ontario called Windsor. So it's you can see the little red star. It's a city that borders between the US and Canada. In fact, Windsor, Ontario faces directly across Detroit. And so in 2016, by chance, we had mosquito surveillance happening in Windsor, and they detected Aedes albopictus. And that was a bit of a concern at the time. And in subsequent years, it has actually established. And what I mean by established is the mosquito has been detected in this part of Canada consecutive years over and over, and in different life stages. So as eggs, as larvae, as adult mosquitoes. And this tells us that this mosquito species is reproducing and having its full life cycle in Canada. So this species of mosquito can transmit a number of different diseases. There are actually over 20 arboviruses, but the ones that you're probably most familiar with are dengue, Zika, and chikungunya. Most probably because we've had recent outbreaks of these diseases in the last decade. So chikungunya, we had an outbreak in 2014 in the Caribbean, Zika virus from Brazil and the surrounding regions in 2016, 2017, and dengue also from last year. So last year was the largest number of reported cases of dengue globally, mainly due to an outbreak in South America. And so this figure that I've got here shows some of the travel-related cases of dengue, Zika, and chikungunya coming back to Canada between the years of 2012 to 2023. What I wanted to point out is in this figure, you can see there's arrows with that points to a histogram bar, and those are sort of high case count years for each corresponding diseases. So you've got Zika outbreak and chikungunya outbreak. And so what this shows is, even though we don't have these viruses circulating in Canada, these are exotic to us at the moment, if there's an outbreak happening globally in other countries of the world, it will inevitably affect the number of cases that we see back in Canada. And so we're not isolated from the rest of the world. What happens globally is going to have an impact on Canadians as well. You don't see dengue here, but I'm pretty sure it's because it's cut off in 2023, but in 2024, we would have seen hundreds, if not thousands, of return cases of travel-related dengue. Unfortunately, these diseases are not nationally notifiable, so we really don't have a way to be able to capture their cases. But we do know that when there is an outbreak globally, it will have an impact on us. And so that's what makes exotic mosquito-borne diseases particularly a public health concern for us, because we're not isolated from the rest of the world. When they come back, when travellers come back with the diseases, we have to manage them in our healthcare system. But in addition, because we do now have an established population of Aedes albopictus, despite being in a very small part of Ontario, we now have the ability that if someone comes back with a travel-related case of any of these pathogens, they could potentially pass it on to someone else who has no travel history if they come back at the wrong time, at the wrong place, I guess you could put it that way. And if there's mosquitoes that are breeding that can transmit these pathogens, someone who hasn't traveled outside of Canada could potentially get sick. And so that's that's one of the most I guess concerning issue with exotic mosquito-borne diseases, is that we have the chance that people can get sick without travel. And because we've never had these viruses enter Canada and circulate locally, it means that it could be quite explosive if we did have outbreaks. So for example, with COVID, it was a naive pathogen that entered the population and it ripped through the population. So when your population is susceptible, you could have these very explosive outbreaks, and that is always top of mind for public health. So we do have traditional mosquito surveillance. So, you know, the good thing is we do have a healthcare system and it is actively looking at mosquitoes. And so this is the Windsor-Essex County Health Unit. They do their own mosquito surveillance. And since the introduction of Aedes albopictus in 2016, they go actively and they're searching on a weekly basis. They're setting out traps to look for mosquitoes, and not just Aedes albopictus. This figure shows Aedes albopictus between 2018 and 2025. And in general, albopictus is detected between the months of July and August in Windsor-Essex. And so they are setting out traps to trap mosquitoes of all different species, and then they're separating the Aedes albopictus. Then they also try to isolate viruses in those Aedes albopictus, and to date, there's been no viruses detected in the Aedes albopictus that's been trapped in the mosquito surveillance program. But it's good to know we do have this surveillance program. It usually runs from late May to mid-October in Windsor-Essex, and many other health units also do have mosquito surveillance. So this is one way to sort of get on top of these emerging public health threats. But with traditional mosquito surveillance, what I have here in this figure is it's actually quite exhaustive. It's quite expensive as well, and the equipment is bulky. So this is just an example from a publication that I've pulled from the Salt Lake City Mosquito Abatement District. This just shows an example. All mosquito surveillance equipment will vary depending on the organizations that's using them. But as an example, here you can see that with this particular mosquito trap, they're using a CO2 oxygen tank. So this is in this panel in A, and it's tucked into this black box that has a motor as well that runs the fan in the mosquito trap. So each of these black boxes, so everything's tucked away neatly in this black box. They get shipped onto this truck, and this truck is probably carrying about 20 or so traps. And they get sent out to different locations, and then this is what they look like. So, you know, they all look different, and this is the particular setup that they have. What I wanted to, the point I wanted to make with this figure is it can be really bulky, and it could be time consuming, and it takes a lot of time because someone now has to go and deliver all of these traps into 20 or so different locations. Usually they trap for 24 hours, then they go back, pack up all the traps, and then sort out all the samples, put them in the fridge, get them to the labs. So traditional mosquito surveillance is not cheap, and it is very time consuming. And so, you know, we are lucky to have very active mosquito surveillance in Canada, but it's not a very, a resource or budget-friendly program to set up. This is just this figure is it shows other types of mosquito traps. So here, these figures A and B are courtesy of the BC CDC. So you might have a different type of mosquito trap here. So this is also with CO2, but they use dry ice in a big thermos, and they dangle the trap near the ground level to collect mosquitoes. And there's usually a battery pack that runs into the trap that has a fan to suck the mosquitoes into the trap. So, you know, some of these mosquito traps are a bit more compact as well. Figures 3S, B and C are ovi traps, and so these ones are designed to have mosquitoes come into these traps and lay eggs. What I wanted to show is that you actually need really large buckets of water, and so setting up this surveillance program takes a lot of time. It's heavy, it's labor intensive, and then you have to dismantle it as well. So traditional mosquito surveillance, it works because it's set up on a weekly basis and it's in real time, but there are some drawbacks to traditional mosquito surveillance. And that's why, you know, we have some sort of citizen science mosquito surveillance programs set up to complement traditional surveillance. So why do we do it? We don't want to take over traditional surveillance. We just want to complement traditional surveillance by giving it extra data points. By capitalizing on the community, we can actually empower and encourage individuals to participate, but only if you can make the method accessible. It's a great educational opportunity to raise awareness in key areas. So you don't want to startle the entire country. There might just be some areas, for example, in Windsor, Essex, where we want to target Aedes albopictus. There might be other parts of Canada where there's other species that are of interest. And so citizen science allows you to sort of be more targeted and raise awareness in key areas. It's been embraced globally and with varying success, but quite well-established programs. And so 2 years ago, one of my colleagues and I set up Team OCS. We thought, why not add our, ring in the hat and see how it goes? And it's also a really great opportunity for unique collaborations with multiple purposes. So I'll speak briefly about the Tiger Mosquito Citizen Science Study. We call it Team OCS for short. It was launched in 2024 by myself and my colleagues, but we've done a huge revamp in 2025, and we've used this year 3 new alternative surveillance methods, but the objectives remain the same. We want to complement, not replace, but complement existing surveillance efforts in Windsor, Essex during the summer months. We want to improve the awareness of mosquitoes and disease risks, and the data that we collect can be used for multiple purposes. So our first method is adult mosquito surveillance. So I had showed you a couple of slides on how big and bulky and varied traditional mosquito surveillance equipment can be. What we've gone for this year is a 3D printed trap. And all the logos at the bottom represent people that I've partnered up with for this part of the study. We have Salt Lake City, the Mosquito Abatement District. They've generously donated these 3D printed mosquito traps. So this trap, which is about 15 centimeters in height, is a 3D printed mosquito trap. Traditional traps can cost up to $200. This trap costs about $20 to make. And so I've built this kit with the donated traps that they gave us. We've got an insulated tumbler for dry ice. We've got a little power bank battery pack. We've got 3 collection tins for the month of June, July, and August, and a mesh collection bag. And I've got 14 of these traps that I've distributed across to Let's Talk Science and the University of Windsor, who we're partnering up with. And each of the volunteers will be able to set up their traps on a weekly basis and be able to collect mosquito samples for us. I also have thrown in our collaboration with the University of Calgary. So John will speak next, and he and I have been in touch about us sending the trap samples to the University of Calgary for identification. And then this information will be passed on to the Windsor-Essex County Health Unit, who are very supportive of the work that we're doing, and they're very interested to have additional data points to supplement their surveillance. The second way that we are doing surveillance through Team OCSSE is homemade OV traps distributed in classrooms. Here's where we actually have citizen scientists that are kids in classrooms from kindergartens to grade 8s. We again have partnered up with Let's Talk Science and the University of Windsor. In fact, I've trained them to go into the classrooms to then go and give a course on mosquitoes and life cycles of mosquitoes and how to prevent mosquito bites, but then to also teach students how to make an OV trap. And then we've sent them home with an envelope, a pre-stamped envelope, and an OV strip for them to collect samples for us and hopefully send them in. I've got the University of Calgary here because, again, we're working with John and his lab, and we're sending those egg samples to be identified at the University of Calgary. And then the health unit will get the data that we receive as well. Now, I made a point about multi-purpose. So with the eggs, I get the identification of the mosquitoes that I will pass on information to the health unit, but I'm also validating my risk assessment models. So I'm using the data to validate risk models to look at the expansion of Aedes albopictus. But then the University of Calgary can rear those eggs and then run vector competency tests on them as well. And John may talk a little bit more about this in his presentation, but it just shows that a collaboration like this, you can have multiple partners, each getting something different from the outputs of the study, but everyone gets something out of it. So the Let's Talk Science volunteers also gets the experience of being a part of science study, and the students get the experience of science outreach and being taught how to prevent themselves from a mosquito bite. So every person or every partner gets something different out of these collaborations. And that's the great thing about citizen science. There is multiple purposes. It's not just one organization with one goal. It can be spread out across different organizations. The third sample that we have is environmental sampling of mosquito DNA. So I'm working with, again, Let's Talk Science and at the University of Windsor. Those volunteers have taken one of these environmental samplers or passive samplers for me, and they'll be going and taking some environmental water samples in different water bodies across Windsor-Essex over the summer, working in collaboration with the University of Guelph, who's currently helping me develop a bioassay to detect the DNA of Aedes albopictus in water sources, and then again working in collaboration with the local health unit, where I'll be able to pass on the data to them, which may help them with their surveillance strategy for upcoming years. So this is my last slide, and I want to sort of end with an analogy. This is the way that I see citizen science and mosquito surveillance is that we have a core piece. So we have the Windsor-Essex Health Unit. They have a public health reason to be doing active mosquito surveillance in that region because of the emergence of Aedes albopictus in the last 8 to 10 years. So they are sort of the key core piece, and there's a common goal here. But in addition, every partner that we have as part of the Team OCS study can bring a little bit into this, whether it's Let's Talk Science and the University of Windsor. They and their volunteers are hugely instrumental in getting into the classrooms, teaching kids in this particular key area of Canada about mosquitoes, how to prevent mosquito bites. Now, we don't actually go and teach kids we don't focus on the disease aspect because we don't want to go and scare the kids, but we're teaching them how to prevent mosquito bites, and we're empowering them and encouraging them to be a part of a science study. We have citizen scientists, so they can go home and talk to their parents and maybe have conversations at the dinner table that they normally wouldn't have about mosquito-borne diseases. So they get something different from this part of the study as well. We have partnerships, valuable partnerships with our academic partners, the University of Calgary and Guelph. And they each also get something different from the outputs of this study as well. We have Public Health Agency of Canada that's leading this study and bringing all the jigsaw pieces together, and everyone contributes something to this. And in general, we have a core mosquito surveillance program at Windsor Essex, but we can add to this, and that's what this figure is trying to emphasize, that everyone has a little small part. We can add this and make it a larger piece of the picture so that we have a better understanding of Aedes albopictus and its distribution in Canada. So I've also put in sneakily our QR code. So if you are listening to this presentation, feel free to scan the QR code. It will take you to our webpage where you can find out more about the study. If you are in Windsor Essex, or you know someone who's in Windsor Essex that would be interested in participating, please share this webpage information with them. And you can also get in touch with me via the webpage email address. So I will stop my presentation here. It was a lot to go through, but I welcome questions during the Q&A, and I will invite John to do his part of the presentation. So I'm going to stop sharing my screen.
Thanks, Vicky. Hello, everyone. So thanks, Rob, for the excellent introduction earlier. I appreciate the opportunity to talk to everyone today. And before I get started on my presentation, I would like to take this chance to acknowledge that I am joining you all today from the traditional territories of the people of the Treaty 7 region in southern Alberta, from the city of Calgary. And the city of Calgary is also home to the Métis Nation of Alberta, Districts 5 and 6. And I have no conflict of interests to disclose today. Most of what I'll actually be talking to you about in terms of the actual work was done by the people beside me in this picture. These are graduate students, postdocs, and undergraduate students, as well as a lab technician that have worked with me for the last few years on mosquito-related projects. And I also wanted to take a chance to acknowledge my excellent collaborators throughout Alberta and in Saskatchewan that have provided a lot of the data that I'll eventually be discussing with all of you, and especially Mike Jenkins from the city of Edmonton and Alex Coker from the city of Calgary, without which the sort of work that I've done on a specific mosquito, as well as connecting me to municipalities and other public health officials throughout the country, it wouldn't have been possible. And part of that is because I'm actually not from Canada. And so my network is quite limited here at the moment. As you guys heard earlier, I did my PhD in Massachusetts, as well as postdoctoral fellowships in various other US states. Throughout my time, though, I was really focused on basic science questions, but I was utilizing public partnerships in order to gather data, but also to try to contribute back in my own research to questions that those organizations may have had. So I worked with the Bermuda Ministry of Public Health to study Aedes albopictus on Bermuda. I worked with the Central Massachusetts Mosquito Control Project on local mosquitoes found near Worcester, Massachusetts. And I worked with the greater community in the Northeastern Mosquito Control Association as Aedes albopictus was spreading through the eastern United States. And now I come to Canada, where we're facing Aedes albopictus moving here as well. I also work with tick testing and pathogen testing programs while a postdoc, and later with different public health entities and public health organizations, in part so that I could access specimens that were critical to my research, but also so that I could help those organizations by bringing molecular methods I know to questions they have. And earlier, Vicky kind of referenced some of this, and that a lot of the roles that I play in projects is that my lab is really well equipped to use genomic methods to understand what species are present and where species are moving to. And throughout my time as a PhD student and as a postdoc, I saw that the benefits that I was gaining and the benefits to my collaborating institutions were really quite vast. I mean, one of the things that I think helps a lot with the collaborating entities is access to new methods or techniques by exposing them to new things from the literature, because that's part of what my job is, to keep updated on those things. I also offer access to expertise and specialists that may not be present. This is both in terms of molecular diagnostics as well as confirmation of species IDs. And this isn't only something I do, but a lot of those of us who have training in mosquito taxonomy, that's quite a specialized skill set at the moment. And so it can be difficult to identify mosquito species. And so having access to an academic with those kind of abilities can be really helpful to public health entities. And it also provides opportunity for evaluating or analyzing procedures or data gathered as part of surveillance. And Vicky kind of referred to this earlier in terms of collecting all of this data from citizen science projects and having the opportunity to understand what species are present and what they could be doing. For me, it gives me access to specimens and data. A lot of my research originated as basic science and understanding where mosquitoes were, what pathogens of a mosquito were there in the environment, and what it was doing to the populations of those mosquitoes. I was actually using mosquitoes as models to study disease ecology, as opposed to actually studying disease transmission to humans. And so it was really critical that I have access to mosquitoes from natural environments, and there's only so much that one person or a small research lab can accomplish on their own. By partnering with various public health entities, I've really gained access to a lot of specimens that I wouldn't otherwise have had access to. And for me, this often means that I can work with people close to home. Many of my research projects end up funded to take me to really cool places that are far off, such as Brazil or Africa. And those projects can be quite important to the kind of research that I'm interested in from a public health perspective, but it does mean that it can often be hard to engage with my local areas and the local communities that I'm part of. And for me, that's actually pretty important because I'd like to be involved in mosquito operations close to home. And so I really need to partner with municipalities. And unfortunately, sometimes the funding that is available for the study of mosquitoes close to home may not be quite what it is, strangely, to study mosquitoes further away. And part of that relates to the mosquito-borne disease and where it's most prevalent globally. The other interesting thing here is that it actually gives me access to practical expertise with people working in the field. And I learn a lot from working with those people because oftentimes we are setting up a limited number of traps in my lab in a limited number of locations. But I work with people who are conducting surveillance for months of the year across dozens to hundreds of sites. This gives me access to a lot of information about what they're seeing on the ground and what works for them. And then also some of the studies I conduct with these folks may have direct benefits to communities or personnels that are undertaking control. And so when I joined the University of Calgary in June 2021, I had interest in establishing a research program that was a little closer to home. And so in late 2021, early 2022, I actually reached out to Alexandra Coker, who works for the City of Calgary in Parks and Natural Spaces, and we started to spitball ideas. And this is kind of how I like to approach a lot of my partnerships, is we come together and we ask, what are we both interested in? And are any of those topics something we might be able to work together on? And so today I'm going to focus on one specific item that Alex brought up, was that in the city of Edmonton, there was a new invasive mosquito which had been found in 2018, but only confirmed in 2021, Culex pipiens. And I'll return to that finding in Edmonton in a moment. And I just wanted to briefly highlight that the work that I'm going to be talking about, the partnership between the City of Calgary and myself, as well as the City of Edmonton, led to a recent publication on the first record of Culex pipiens in Alberta, as well as discussions on what this could mean for public health in the province. Okay, so before I get into this sort of expansion, though, I should introduce Culex pipiens. Earlier, we heard about Aedes albopictus, which is a major invasive vector. Culex pipiens is arguably the second or third most important invasive species globally. Culex pipiens is an important vector for zoonoses. It's one of the main vectors of West Nile virus, circulating that virus in bird reservoir hosts, but also because it will occasionally bite humans and other mammals, it can transmit West Nile to us. It's part of a species complex where there are many species, subspecies, and ecotypes that occupy different ecological niches, which gives this species a lot of adaptability and a lot of ability to survive in really complex and different environments. That also leads to it being highly adapted to urban environments. This is a mosquito that will make use of the unintentional habitats we place out in our yard. Everything from buckets, rainwater collection, tires, even just runoff from our houses. And so this is a globally invasive species that is well adapted to living right alongside humans. And for most of, so its global distribution is quite large throughout the temperate regions of the world, through North America, Europe, Asia, and Northern Africa, as well as into some parts of Australia. Looking specifically in North America, though, for most of our knowledge of the species, we believe that it was located predominantly in eastern Canada, as well as along the British Columbia coast and into central BC, but that the prairies were largely unaffected by this mosquito. The gray band was the presumed distribution as the early 2000s, and there have been new reports from Newfoundland, Labrador, etc. Important to note for this presentation, though, extensive surveillance in Alberta through the 2000s and 2010s suggested that Culex pipiens was not found in Alberta. This was true of Saskatchewan as well. However, habitat suitability modeling predicted that over the next 30 to 60 years, there would be a 5-fold increase in suitable Canadian habitat for this species. This is also true of Aedes albopictus, which is one reason why we're concerned about that species and monitoring it in Windsor with our partners. And new habitat suitability estimates actually suggested that parts of Alberta were already moderately suitable, likely due to urbanization and climate change. And that's going to play an important role in a second. So in 2018, mosquito surveillance work found a new mosquito in Edmonton that the surveillance workers had not previously seen. This mosquito closely resembled a mosquito that that mosquito control worker had previously seen in Winnipeg, but had not seen in Alberta yet. And at first they thought this mosquito might be a different Culex species, Culex restruans. It was not until 2021 that that mosquito was barcoded, and it was determined that in fact Culex pipiens was found in Edmonton. I've highlighted the 3 locations that Culex pipiens was found in Edmonton in 2018, and you'll see that by 2018 it was actually found all over the city and outside the city. Within a few short years, Culex pipiens had been found at virtually every trap within the greater Edmonton area. As a result of this, there was rising concern throughout the province that Culex pipiens could be elsewhere. However, because of typical mosquito surveillance occurring in the province, we weren't really sure where it was found. And that's because this mosquito is highly adapted to urban environments, and a lot of Alberta's mosquito surveillance programs are largely geared towards sampling mosquitoes that are found throughout more floodwater habitats. So this presented an interesting challenge for surveillance entities within Alberta. And this is part of where I came in. So I started talking with my colleague Alex Coker, and she had 2 big questions. She wanted to know, where was this mosquito found, both in Calgary, but also elsewhere in Alberta, and then what was it doing here? There's a third question I won't have time to get into, which was, where did it come from? And that's kind of a more interesting question we're still trying to figure out the answer from. In order to address where this mosquito was, we used larval surveys, bucket overtraps, and adult traps, which Vicky kindly introduced to us earlier, so I won't spend too much time on them. And we sampled with larval surveys throughout the city of Calgary. I haven't marked those sites on this map because we sampled about 40 different locations. Amusingly enough, in 2022, when we first started our sampling, we did not find a single Culex pipiens with larval surveys. At this point, we had not detected the mosquito, and so we wondered if Culex pipiens was in Calgary at all. Well, following this, I contacted a colleague of mine back at North Carolina State University who had worked with Culex pipiens throughout his PhD, and he suggested we try bucket ovitraps. So we placed bucket ovitraps first in one location in Calgary. Full disclosure, this was actually my porch patio of my house in Calgary. And within a few days, I found Culex pipiens eggs. A couple of other bucket overtraps were placed at different locations throughout the city, and a couple of them turned out positive. Then going back to sampling of adult traps, which were placed in July, we were able to find more Culex pipiens. However, because there were 90,000 mosquitoes collected in Calgary in July, we took quite a long time to get through all of those mosquitoes. And out of those 90,000, we only found 30 adults. So at this point in time, we felt that maybe Culex pipiens was somewhat rare within the city. The next year, we decided to get our bucket overtrap monitoring program started much earlier. So working with the city of Calgary, we placed buckets throughout about 3 dozen park depots and other locations throughout the city. Every single one of them came back positive. in 2023. Our first larvae, we actually were able to collect from habitats on July 2nd. And by the end of the summer, every single bucket overtrap in 2023 was positive, and virtually every adult trap, CO2 trap we placed was also positive. In other words, in a single year, the positive sites we gained essentially exponentially exploded throughout Calgary, and the mosquito was found all over the city. This has maintained through 2024, and we've already seen it in 2025, which means we think we actually sampled the beginning of the arrival of this mosquito in 2022. We reached out to other municipalities, such as Red Deer, in Olds, and Lethbridge, and in 2023, all of those municipalities located Culex pipiens in Alberta as well, indicating that this mosquito was likely widespread now across southern Alberta. In addition, in Lethbridge, colleagues of ours at the Agriculture and Agri-Food Canada looked at older trap contents of mosquitoes that had been unidentified or could not be identified at the time because they didn't resemble any mosquitoes found in Alberta, and they found multiple samples from 2018 that looked like they might be Culex pipiens. This means that it's possible that Culex pipiens was established in the province and within Lethbridge prior to our knowledge of it, and perhaps before it was found in Edmonton as well. So the next question that we had was, what is this mosquito doing here? And this relates to kind of, once you find an invasive mosquito, that is problematic if you know what diseases it transmits or what it might do elsewhere, but you really need to know what it's doing where it is now in order to understand what influence it may have on public or veterinary health. And as an academic, I'm often interested in sort of more basic questions. I love to study population genetic data to figure out where species are from, things like how is it adapting to our cold. Those kind of things have public health implications. Other times they don't, but my collaborators are luckily interested in those things as well. But from a more general perspective of what matters to Calgarians, it's more about where this mosquito is and what it's doing. And so we contacted our colleagues in Edmonton and drew on their expertise and experience in the field of observing where they were finding this mosquito to try to characterize what it was doing in Calgary. In Edmonton, they noticed this mosquito was making extensive use of catch basins for larval habitats. In fact, they can find larvae in catch basins into January, suggesting that this mosquito may actually be overwintering in catch basins or at least developing well into the winter. They've broken ice and found mosquitoes alive underneath it, which is quite astounding to me. Interestingly, in Edmonton, almost all of their catch basins hold water and for quite a long time, even through most of the year. But in Calgary, there's only a handful of neighborhoods that have catch basins that hold water. Almost all of them drain immediately due to flood concerns. So what we did was we targeted 2 of those new neighborhoods shown here with circles, and we established a catch basin monitoring program with the city of Calgary. We sampled both locations using a modified fishnet on an extendable pole, which you can see one of our technicians using there. And we found Culex pipiens at both sites, but they were not particularly abundant and they were not found during all collections, suggesting that although Culex pipiens was using these habitats, it didn't appear that they were using them at the level that was occurring in Edmonton. Nonetheless, we decided we will continue this program this year, focusing on one site and using a different method. We're actually going to be using a more intensive sampling method where we remove all of the water within a catch basin and then filter it to find any mosquito larvae. This is because we're concerned that our method of sampling larvae may have unintentionally caused us to miss larvae that were present. If these catch basins prove to be a major potential breeding area, this may allow the city of Calgary to make different decisions during urban planning to help head off the potential habitat for invasive mosquitoes such as Culex pipiens to utilize aspects of urban development. But what are the adults doing and where are they found? We know almost nothing about Culex pipiens and what it's doing in Alberta. We don't know where the adults are active, when adults are active, what they're doing in terms of who are they feeding on? Is it humans, birds, etc.? How are they surviving the winters and what pathogens are they associated with? And all of these questions require us to find adults. So most of our focus was on using traps and other methods such as what Vicky mentioned earlier. So bucket traps are good for a presence of a mosquito, but for us, it's more important to catch adults because we can then do things with those adults, such as, look at when we found them. We know adults are active at that point and host seeking. It also lets us know if there are any pathogens found in that mosquito, although I won't be talking about pathogens today. There are many different types of traps, as Vicky mentioned, and I'll quickly mention that this BG Sentinel trap is just a CO2 trap. So this is the same kind of trap that Vicky mentioned earlier, but it's just a slightly different model. The Gravid trap has this hay-infused water, which simulates a mosquito breeding source, and then mosquitoes get sucked up into the tackle box. And then we used a modified resting trap, which is supposed to create an environment for mosquitoes to hang out in after they've blood fed. And we used an aspirator, which essentially is where you sample in an area, specific area near where you think mosquitoes may be resting, and you try to suck them up. We sampled across a number of various sites throughout Calgary, which were natural or non-urban sites. I'm highlighting the zoo specifically because unfortunately, in 2023, there were 2 penguins that died of avian malaria, which is a veterinary pathogen that is transmitted by Culex pipiens. So we're a little worried that this mosquito could be driving disease outbreaks in animals as well. After 15 weeks of field work, we collected 4,307 mosquitoes. Culex pipiens was first found in mid-July in our adult traps. Now, we only found 127 Culex pipiens across those different 5 sites. Now, one of those sites we didn't find any in, so really it was found across 4 sites. But note that Edmonton itself catches more than 1,000 Culex pipiens per month, and we found only 125. Now, to be fair, other sites in Calgary do catch more Culex pipiens than us, but nonetheless, it remains the fact that Culex pipiens is still less common in Calgary than it is in Edmonton. Between our different collection methods, our CO2 trap, our BG Sentinel, collected the overwhelming majority of mosquitoes, but the majority of those mosquitoes that we collected were Aedes mosquitoes. We were specifically interested in Culex pipiens, and we did 49% of all of our Culex pipiens were collected in those BG Sentinel traps. BG Sentinel traps also collected 5 different genera of mosquitoes and collected mosquitoes every time they were placed. Our Gravid traps collected 72% of mosquitoes and collected slightly more as a percentage of Culex pipiens. The modified resting trap, however, only collected male mosquitoes, which is a little too bad because we were hoping to use it to find out what mosquitoes we're feeding on. And our aspirator only collected Aedes mosquitoes, probably because they were trying to bite us as mammal hosts. So moving forward, we're going to be giving up those 2 traps. But the other aspect of adult surveillance is it gives us access to a bit of phenology. When are we finding mosquitoes? We found mosquitoes at our urban sites first on July 11th. Next, we found it at the Calgary Zoo on July 18th. Sorry, these are Culex pipiens mosquitoes. And that's a semi-urban site in Calgary. It's within a highly developed area, although there's a lot of tree cover, which sometimes makes it classified as not quite urban. We then found Culex pipiens at the Inglewood Bird Sanctuary, and we never found Culex pipiens at our natural site outside the city. If we look at the abundance of this mosquito, there was a spike in the number of Culex pipiens collected in late July or early August. This corresponds with when Edmonton sees the most Culex pipiens. And we also saw a second spike of Culex pipiens towards the end of the month, which may indicate a second generation of this mosquito was active in Calgary. In terms of where we found the most pipiens, we did find the majority of them at the Calgary Zoo. And then the second most were found at our urban sites, which were just park depots within industrial or residential areas. Our next big question related to Calgary. As a lot of you know, Calgary gets quite cold, -40 sometimes. We don't actually have that much snow cover though, but it's pretty cold pretty year round. And this is one of the furthest north locations you can find Culex pipiens. So how is this mosquito surviving these winters? We know that the mosquito is called a house mosquito for a reason. In other locations, it's been known to go into people's houses to overwinter, as well as to use other man-made structures. So what we did was we set up a citizen science campaign called Bite Back, where we engaged the community across Alberta to mail us mosquitoes they found in their homes. Mosquitoes that are overwintering often look dead because they enter a state of suspended animation that is called diapause, and they'll oftentimes fall over during diapause and simply be on their backs, which honestly is kind of hilarious. But so we just asked Albertans to send us their mosquitoes. We received mosquitoes from Edmonton, Calgary, and Lethbridge, and Culex pipiens was the most abundant species we found, and we found it across all 3 municipalities. We also got a whole lot of midges, which weren't really our target, but may indicate that we have some public education work to do. This year, we've started a new citizen science campaign that was inspired by the Soghigian Lab at Acadia University. We wanted to know what mosquito adults were feeding on people or being attracted to them. Ideally, before they bite people, but we wanted to know what mosquitoes were being attracted to people. And so working with the city of Calgary, we promoted our Skeeter Seekers campaign, where we asked residents to take a mosquito collection kit, which is just a little vial with a bag and a little piece of paper that they can write information on where they were when they collected the mosquito, and to either smash a mosquito that lands in them or collect the mosquito, put it in the little vial, put it in the bag, and drop it off in one of our coolers. We've already received about 30 samples after about a week of collections. And so residents have been really interested in this. We received a lot of really positive press, and we're hoping that this will help us understand what mosquitoes are out there biting people. So what we know so far is that Culex pipiens is widespread throughout southern Alberta. It's rapidly expanded throughout Calgary. Culex pipiens uses catch basins, but in Calgary, it may be less than Edmonton. We need to do a little more work before we conclude that. Culex pipiens activity appears to peak in early August, but we have limited data so far. This is consistent across the municipalities that are sampling adult Culex pipiens. This might indicate that the greatest risk for disease transmission of things like West Nile virus could be in August in Alberta, August or September. Culex pipiens is overwintering inside our houses. Honestly, it's probably inside parkades and other structures. So one of the things my PhD student will be doing this winter is going to public areas where we can sample against corners and things like that to look for mosquitoes, such as in parkades owned by the city of Calgary. And one other note that I think is one of the biggest things that this collaboration has led to is that the collaboration with the city of Calgary has created a workshop series that I run every year now, or I'm the coordinator for. And the City of Calgary personnel that are actively engaged in mosquito management also take part in helping run this workshop. They're teachers on the mosquito control and surveillance end of things. And we sponsor a vector biology workshop that's designed for anyone with a basic education in biology that has an interest in learning more about mosquito sampling, surveillance, or public health risk. It's 3 to 4 nights, depending on the year, in late May or early June at the Biogeoscience Institute in Kananaskis, Alberta. It's less than $500 all inclusive, and every year we hope to have scholarships available. So if you're interested at all, feel free to reach out. We often also offer continuing education credit for those individuals that are interested. And over the 4 years we've run the program, we've trained more than 80 individuals from academia to municipalities, as well as from public health agencies at the provincial and federal level on mosquito surveillance and control. And so with that, I just wanted to quickly acknowledge all the people who actually generated any data. And I think we could take any questions that anyone might have right now.
All right, thank you, Victoria and John, for sharing your expertise and knowledge on mosquito surveillance and your many partnerships and collaborations. We will now begin our discussion period. So please send in any questions you have using the Q&A tab. Everyone is welcome to participate. We'll try to address as many questions as possible. So just to start us off, we had some questions during the registration. This is for both of you. How has climate change made mosquito surveillance more or less difficult? What challenges do you foresee facing mosquito surveillance in the future?
Do you want to, I can go first, or it's up to you, Vicky.
Sure,
So
go first.
Okay, so I'd say, okay, so I guess there's multiple layers to that question. The way in which climate change has significantly altered mosquito surveillance is it is changing the species found in different locations. So I touched on this very briefly, but as an example in Alberta, Culex pipiens is the only species we have here right now, thankfully, that is well adapted to urban environments. Most of our mosquitoes are what we would typically call floodwater or floodplain mosquitoes. Those mosquitoes, their eggs are laid in flooded areas where they hatch, and the adults can fly great distances in order to find human hosts. What that means is oftentimes surveillance is outside of downtown cores or outside of downtown residential areas to track the mosquitoes that are coming into places to feed on people. Culex pipiens, though, is different. Culex pipiens lives in our backyards, quite literally. It uses bird baths. It uses water cans, which are watering cans for flowers or things like that, flower pots. anything we leave out in our yard that gathers water. And this is true of Aedes albopictus as well. And so these mosquitoes are incredibly well adapted to living really close to us. So that changes a little bit about what species are found. This also changes some of the ways we have to train people because mosquito distributions are shifting so quickly. It's a lot harder on us training the surveillance technicians that need to be identifying species. Because a lot of times the easiest thing to do if you get someone new into the lab and you're asking them to go through a trap from a locality is to say, these are the species here. This is the key to the species that are here. And we're going to use this key. Unfortunately, when we do that, we miss potential species distribution changes. Because if a species is now entered into our area due to climate change that wasn't there before, it may be missed. And this is a little bit of what happened with Culex pipiens in Alberta. It also changes the potential viruses and other pathogens that could be present. So as mosquitoes move around, so do their pathogens. That can then change which species could be vectors of disease. So oftentimes we focus on species like Aedes albopictus and Culex pipiens because we know they transmit diseases elsewhere. But as they move into new locations, they can become engaged in disease transmission in unexpected ways. An example of this is a California serogroup virus member lacrosse virus. In Eastern North America, where the primary vector of lacrosse virus was Aedes tricereatus for a very long time. Now, this mosquito is considered a forest mosquito. It will use things like tires in artificial habitats, but it mostly is found inside of forests and not so much in urban areas. However, Aedes albopictus is very competent for lacrosse virus as well, and there is now rising concern in the southeastern United States that Aedes albopictus could become a secondary vector of lacrosse virus or potentially more and transmit that virus to more people. And so that opens up this new mosquito entering an area could change disease dynamics tremendously. I forgot the second half, so I'll just leave Vicky or offer any more on this topic or on the second half of that question.
Victoria, did you have anything to add?
I was going to say, John was so comprehensive with that response. So I don't really have much to add other than I think in addition to what John mentioned was that we're going to expect to see longer and earlier mosquito seasons. So that's going to have an impact on, as I mentioned, it's really expensive to do traditional mosquito surveillance. So it's going to have an economic impact on public health to try to keep on top of the mosquito surveillance system. There is that expanding geographic range of the distribution of diseases. So not only are we having to survey longer periods of time, but even larger geographic areas as well. And it is already limited at the moment. Like we are probably at capacity in terms of what we can fund for mosquito surveillance. And so climate change will just cause things to be a bit more difficult, and we will have to make some hard decisions on, you know, how we fund certain programs.
Well, thank you. We are short on time, but I will ask one more question that we had from our audience. This comes from Che. How does this research fit with the international malaria landscape? If your research applies.
So I mean, I can briefly answer. So for the most part, because of where malaria transmission occurs, my specific research typically doesn't overlap with malaria as much as with the Aedes disease vectors, which are more invasive. But a lot of the general methods in terms of trap testing, and I didn't talk about this at all, but Vicky mentioned briefly, like competency of different species and these kind of things, it does matter for malaria-related research as well.
Yeah, similarly, I guess my study is focused more on Aedes albopictus. It's one of 3 main genuses of mosquitoes that can transmit viruses and pathogens in general. And so it doesn't really focus on the species that can transmit malaria. But there are certainly cross methods and even the way that we trap mosquitoes and do mosquito surveillance. That lessons learned from these citizen science studies can be transferable to other mosquito surveillance programs for other mosquito species, but I don't necessarily deal directly with the vectors of malaria for my study. But that's not to say that we can't expand in the future, but just at the moment, we don't focus on that.
Yeah.
Thanks. Yeah. So that concludes our discussion period today. I would just like to thank everybody for joining us for this seminar of surveillance advances, and to our speakers, John Soghigian and Dr. Victoria Ng, for their fantastic presentations. Just a reminder that the recording for today's seminar and the presentation material will be accessible for viewing on NCCID's website in a few weeks. Also, please take some time to complete our survey for this presentation. You can scan the QR code on this slide to access the survey, or look for a link that will be emailed to you shortly. In the post-seminar survey, you will also be able to request a letter of participation for your attendance today. Please join us for our very last seminar of the 2024 to 2025 season on Tuesday, July 29th, 1 to 2 p.m. Eastern Time, or 12 to 1 p.m. Central Time. If you have any more questions about the Surveillance Advances seminar series, please visit our webpage at nccid/Surveillance Advances Seminar Series for more information. That concludes today's seminar. Thank you all for joining.